The human digestive system consists of three main parts: the foregut, the midgut, also known as the small intestine, and the hindgut. The stomach is the final part of the foregut, where proteins are digested. From here, the contents of the intestines proceed to the small intestine. Here the enzymes in the intestinal juice and pancreatic juice break down the essential nutrients, that is, proteins, carbohydrates, and fats. Intestinal juice is produced by the glands of the small intestine, while the pancreas secretes pancreatic juice. The bile, produced by the liver and stored in the gallbladder, is also emptied into the small intestine. The bile does not contain digestive enzymes. It emulsifies fat drops to aid in their digestion. The outer layer of the small intestine is the serous membrane. Under this layer, there are layers of longitudinal and circular smooth muscle. The synchronized motion of these two muscle layers ensures the peristaltic motion of the stomach. It mixes and propels the contents of the intestines. The digestive system is lined by a mucous membrane, which consists of a thin layer of smooth muscle, connective tissue, and epidermis. The glands that secrete the intestinal juice are embedded in the mucous membrane. Circular folds increase the absorptive surface of the small intestine. Microvilli and villi further increase the absorptive surface of the small intestine. Carbohydrates and amino acids released with the digestion of proteins are absorbed into the blood vessels, while lipids are absorbed into the lymph vessels. The liver is the largest gland in the human body. In adults, this reddish-brown organ weighs about 1.5 kilograms. Its best-known function is bile production, but it also plays a key role in several metabolic processes. The hepatic portal system is located at the lower part of the liver. The hepatic portal vein, the hepatic artery, and the nerves enter at this point, and the lymphatic vessels and common hepatic bile duct exit here. The liver receives its blood supply via two blood vessels. The hepatic portal vein ensures that nutrients that are absorbed by the gastrointestinal tract reach the liver, while the hepatic artery provides the liver with oxygen. The hepatic vein is responsible for carrying blood away from the liver. The liver consists of two lobes which contain numerous hepatic lobules with a distinctive structure. The hepatic artery, the hepatic portal vein, and the interlobular bile ductules form a portal triad embedded in the connective tissue surrounding the hepatic lobules. Blood enters the central vein from the hepatic portal vein and the hepatic artery, which run between the cells of the hepatic lobules. Bile canaliculi, located between the hepatic cells, collect bile produced by the cells and carry it to the interlobular bile ductules. One of the functions of the liver is to neutralize toxic substances that are absorbed in the intestines, as well as to inactivate viruses and bacteria that are found in the blood. The liver converts these into harmless substances. It also plays an important role in the metabolism of macromolecules, including lipids and proteins. The liver builds these macromolecules from their constituents according to the needs of the body. The liver is also essential in maintaining the body's glucose level, as it is capable of storing energy in the form of glycogen. When the blood glucose level drops, the liver breaks glycogen down into glucose. Bile is carried to the gallbladder via the bile ducts. While stored in the gallbladder, bile becomes concentrated and is then released into the duodenum, where it disperses fat drops in food as it is being digested. As a result, the surface area of the fat drops increases, making it easier for enzymes to break them down.